Complex genetics are systems of inheritance that don't follow the dominant recessive system that Mendel found in his pea plants. You have to remember that meiosis and fertilization occurs the same way, but it's the relationship among the alleles that is not the same. For example, instead of a heterozygote having the same phenotype as the homozygous dominant, like it does in Mendel's system, the heterozygote may have its own phenotype. This would be like a red and a white flower mating and producing pink flowers. There can also be more than just two alleles that Mendel found. Blood types, for example, have more than just two alleles in the human population. It's these systems of inheritance that most human genetics follow. We have very few traits that are dominant recessive. Well, I mean, very few traits that matter. This is the first non-Mendelian system, and it's called incomplete dominance. Just like Mendel's system, there are two alleles, but they interact very differently. It's tempting to apply the words dominant and recessive to the system, but those words are not true. In fact, this picture here uses the capital and lowercase r and r, but don't be fooled. We could use two different letters here and it really wouldn't matter. This is because the heterozygote has its own phenotype in this system of inheritance. Unlike Mendel's genetics, where one of the alleles does not mask the appearance of the other allele. The heterozygote has a phenotype that is intermediate to that of the other two phenotypes. In this example here, which is a good illustration of this, there's an allele for a red flower, and when you have two of those alleles, you get a red flower. There's also an allele for a white flower, and when you get two of those alleles, you get a white flower. However, when you get one of each of these alleles, you get a color in between red and white, meaning pink. Humans have a few systems of incomplete dominance. This is true for the genes that control cholesterol management in your body. Your cells, especially the ones in the liver, make proteins that go into the cell membranes and attach to LDL, which is a type of cholesterol. It's a low-density lipoprotein. As shown in the picture on the left, if you get two capital H alleles, you make these proteins and they bind the LDL cholesterol, removing them from your blood. Right? And that's great because then the cholesterol is not floating around in your blood and potentially clogging your arteries. There's also another allele referred to as lowercase h in this picture. But again, don't be fooled just because we're using the capital and lowercase letters. We are not indicating dominance or recessiveness. If you inherit one of each of these alleles, you still make the protein, but you just don't make a lot of it. As shown in the picture in the middle, you have receptors. They bind the LDL cholesterol, but you will still have an overabundance of LDL in your blood. Luckily, this can be managed with a healthy lifestyle, good food choices, and exercise. In fact, many people who are heterozygous with this condition don't know it until they get a little older. Unfortunately, if you inherit two of the lowercase h alleles, you have no ability to bind cholesterol. This can create problems in young children and adolescents as they are accumulating cholesterol buildup in their arteries from day one. These people don't make the receptor at all, and thus they have no ability to bind the free-floating LDL in the blood. This can be managed if you know you have it, but most people are not tested for this disease at birth unless you're certain that someone in your family has it. Sickle cell anemia is also incomplete dominance. It's often incorrectly referred to as dominant and recessive, but it is actually incomplete dominance. There are two alleles for the shape of hemoglobin protein. If you receive two alleles that make the hemoglobin correctly, your little red blood cells are like those round ones in the picture there. This roundish cell has a shape called bioconcave, and this is the ideal shape for carrying oxygen and carrying as much of it as your red blood cells can. People with two of these alleles do not have sickle cell anemia. There is also an allele that can make hemoglobin, but the protein is a little misshapen. This incorrect shape results in the red blood cells being sickled cells or crescent shape. That shape is not ideal for carrying oxygen. It can carry it, but it just can't carry as much of it as those bioconcave shapes. The amount of oxygen it carries is also not enough for the tissues of the body. Thus, this results in anemia, which is a deficiency of red blood cells. There are many types of anemia, but this type is genetic, and this type affects the shape. There are no people in the population with two of these alleles. Fetuses with two diseased alleles do not make it to birth. In early development, the cardiovascular system and blood develops pretty early, and it's incapable of serving the growing fetus's tissues. However, there are people who have one of each of these alleles. These heterozygotes express the sickle cell anemia disease. And many times, healthy people with this disease may not feel the effects of it until later on in life. You might expect that these people have a 50-50 split of bioconcave sickled red blood cells, but the immune system actually tips that balance to 40-60, with the bioconcave cells mounting to 60% of the circulating red blood cells and the crescent sickle-shaped cells resulting in 40% of a person's blood. Sickled red blood cells are incapable of being infected with malaria. In regions of the world where malaria is common, people with this disease live longer and reproduce more. People with the bioconcave red blood cells that get infected with malaria can die. These days, thanks to modern medicine, that's not exactly true anymore. But the people with 100% bioconcave cells are at a greater risk of contracting malaria and dying from it. This map here shows you that in areas with high incidence of malaria, there is also a high incidence of people with sickle cell anemia. So sometimes it pays to have this disease. But in areas where there is no malaria present, it pays to have bioconcave cells. It's going to depend on the surrounding environment. To circle back to why I said that this disease is considered dominant and recessive, this comes from the fact that there are only homozygous dominant and only heterozygous people in the human population with the disease. And this incorrectly prompts people to say that there are dominant and recessive disease types. That's not true. There's actually dominant and heterozygous disease types. I seriously can't remember why I put this picture of the rabbit here. I left it in there because it's cute and fuzzy. Probably soft, too. Regarding genetics, there is a situation where you can have more than just two alleles. In Mendel's genetics, we had only two variants of a gene, the dominant allele and the recessive allele. In multiple alleles, we have three or more alleles. Blood types is a great example of multiple alleles because in humans there are 3 alleles that generate blood types. This results in 4 different blood types, A, O, B, and AB. The ABO system of inheritance for blood indicates what types of proteins are inserted into the cell membranes of the red blood cells. For example, people with type O have no proteins in their red blood cells. People with type AB have 2 different types, A and B, and type A and type B, they're a little bit more complicated to explain. The system of inheritance that determines if you are positive or negative, regardless of your ABO designation, is a completely different system. In fact, being type O has absolutely no bearing on whether or not you are positive or negative. These 2 traits are completely unrelated, kind of like inheriting the ability to metabolize cholesterol has nothing to do with your eye color, or at least I don't think it does. I'll have to look that up. When we have 3 alleles, we usually have a superscript, as you see in this example of blood types. The uppercase I and the lowercase I tell us that there are variations of the same gene. There are 3 variants here. One allele gives you the power to have the A protein, noted by the little yellow triangles in the picture, and another allele gives you the power to have the B protein, noted by the little yellow spheres in the circle in this picture. What then is the lowercase I? Well, it's actually just a placeholder. This is an allele that basically says, don't make a protein. In addition to being a case of multiple alleles, blood types are also what is called codominant. This means that the heterozygote has a phenotype that expresses both alleles that it inherits. This picture here of the fish is like the perfect example of this. If a red and a blue fish mate, they don't create a purple fish. That would be incomplete dominance. Instead, they produce a fish that has both red and blue coloring. This means that both alleles are expressed and coloration is codominant. Blood types are also codominant, and this is exemplified by the type AB people. They have both the A protein and the B protein expressed in their little red blood cells. The next 2 slides are the opposite of each other. Pleiotropy is a situation where one allele, something like a master allele, reaches out and influences other genes. Human albinos are an example of this. There is a gene for albinism. When it gets passed on, it reaches out and turns off other pigmentation genes. Even though these people might have the ability to make the skin pigment melanin, they don't. That gene is prevented from being expressed. It's as if the albino allele has turned off the other genes. This goat here is actually not an albino. It's just a recessive white goat. Many years ago, there was a population of white deer in the Seneca Depot over between Seneca and Cayuga Lakes. These were just white deer in the depot, and they were incorrectly referred to as albinos. White is just a recessive coat color in deer. Opposite to the master gene that affects others, polygenics is when there are many genes that influence one trait or character. Many pigmentation genes would work together to create eye color, hair color, skin color. This is why there are so many different variants of these pigmentation states. This also creates a fluid spectrum of phenotypes that can be sometimes difficult to tell apart since they're all just slightly different from others. The next slide gives you a really good visual on skin color as an example of polygenics. This isn't exactly how skin color is expressed or inherited, but let's use this as an example. It's a great example. Let's assume that you have 3 genes that work together to create skin color. Each gene has 2 alleles that interact in a dominant recessive fashion. One allele says to make melanin, and the other allele says not to make melanin. Again, we have 3 genes, so that means that you can accumulate a total of 6 alleles that combine to make your skin color. This huge Punnett square. It could use letters, but I love that it gives you the visual of the 6 alleles, all of which can be black, making melanin, or white, don't make melanin. Look at how this creates a huge spectrum. The most common combination of alleles is having 3 that make melanin and 3 that don't make melanin. The most infrequent combinations are all recessive or all dominant. This relationship creates a bell curve of frequency of skin colors. This is like a distribution of grades in a class where very few people get hundreds, very few people get 60s, and most everybody is in the 80s range.